Hello and welcome to today's special GeneWiz Week webinar, Creation of Hypomorphic Alleles for Functional Gene Studies. I am Dr. Sumit Kumar, Senior Product Manager at GeneWiz, and I will be moderating today's webinar. I am very pleased to introduce today's presenter, Dr. Sergey Juranovic. Dr. Juranovic received his BSc and MSc from University of Belgrade, Serbia. He received his PhD from Eberhard Karls University and Max Planck Institute for Developmental Biology at Tübingen, Germany. There, he worked under the supervision of Dr. Andre Lupus, investigating the evolution of protein structure and function from ancestral set of peptides which possibly emerged as RNA cofactors in the primordial RNA world. He also studied the structure and mechanism of action of AAA ATPases, focusing on the regulation of protein degradation by proteasomes and associated ATPases. He went on to determine the structure and organization of AAA ATPases that form the 19S subunit in proteasomes. In 2007, he moved to Rachel Green's laboratory for his postdoctoral studies to study the mechanism by which microRNAs control gene expression at the level of translation. He first proposed and then described a novel kinetic model that explains sequences of events that define the mechanism of microRNA mediated translational repression. In 2013, he joined faculty at Department of Cell Biology and Physiology at Washington University School of Medicine in St. Louis. His research group is interested in understanding molecular connection and mechanics of targeted mRNA regulation both by microRNA pathways, components, and other post-transcriptional regulatory mechanisms dependent on mRNA sequence features and RNA binding proteins. If you have any questions for Dr. Juranovic during the webinar, please enter them into the Q&A section and we will try to address as many as possible at the end of the session. With that, please welcome Dr. Juranovic.
Thank you, and thank you for the invitation for the webinar, as well as thank you for the opportunity to present our work on hypomorphic alleles of genes. Before I actually start talking about our technique, I just want to quickly remind listeners about the defects in gene expression that arise from the gene expression amounts. And as we all know, these defects are connected either with the growth defects in chromosomes that can be either amplification of additional chromosomes or missing chromosomes in the organisms. Both actually have as final outcome reduction or amplification in gene amounts that are coming from these chromosomes. Very similar phenomenon is a phenomenon of haploinsufficiency, where in the diploid organisms, loss of one copy of the gene can cause different diseases because the amount of gene product that is coming from just one allele is not enough to suffice normal cellular homeostasis. A lot of these genes that are haploinsufficient are actually important for like cellular homeostasis, and problems with haploinsufficiency rise in all kinds of diseases, including cancers. The problem of gene amounts was discovered really early by American geneticist Herman Muller, who actually found that mutations can actually create changes in the gene amounts, and he claimed these terms as amorph, hypomorph, hypermorph, antimorph, and neomorph, depending on what mutations do to the genetic situation of the gene product. And really, what I really want to focus today is things that describe hypomorphic mutations, which are actually known to be a partial loss of gene function. So these are not complete loss of gene function, but rather reduction in the either levels of protein and RNA that are coming from the gene, or these are the reduced functional performance of the gene. And actually, Hermann Müller, for his work on mutations and effects of mutations on genetics, got the Nobel Prize in medicine in 1946. I quickly want just to depict what hypomorphic really means on this image of a mouse with the mutations in RIRI gene that were found basically that amounts of RIRI protein actually quickly correlate with the size of mice eyes, as can be seen from these images. So the full wild type levels of RIRI gene would give perfectly normal eyes of the mouse, while partial reduction or even bigger reduction in the amounts of RIRI gene products would reduce the size of the mouse. For the works like this, really, the things that we had to do previously would be do the big genetic screens and then found mutations that correspond to phenotypes that we've seen on these images. However, a lot of people in the science and in genetics were wondering how we can actually quickly assess how the reductions in the genes can influence its function. And really, if we want to think about that, we really need to go back to the central dogma of gene expression and look at 3 layers of regulation that usually we study. And they are depicted here as transcriptional regulation, post-transcriptional regulation, and post-translational regulation, depending what we really regulating. Is it a transcription, meaning amount of RNA that comes from the DNA of the gene? Is it a protein amounts that are coming from the RNA during the translation, or is it stability of the protein by post-translational regulations? And these all concepts were used actually to control gene outputs through history and through development of approaches to actually regulate gene expression. However, we should really focus on the current problems that we have in the field of basically applying these tools to genes. So the typical problem here depicted with this comic from Nature magazine and called Adventures in Synthetic Biology is that gene regulation, even though we sequence so many genomes, we still really are reading the code, meaning we are still really looking what are the motives that are used to regulate genes. In this respect, we really need to say that while a lot of things are actually shared between similar organisms, every organism is really specific, and a lot of things that we find can work in one organism, but they don't necessarily work in the other or even really related organisms. And this just shows the complexity of the tree of life that when we are thinking about genetic terms. Now, in very schematic scheme, we would like to point out that even between bacterial and eukaryotic gene expression, when we consider the conserved boxes that we are thinking about using for the regulation of gene expression, there is a difference actually between these conserved boxes. The promoter strength, the promoters that are used to regulate promoter strength are different between bacteria and eukaryotes. The strength of ribosome binding site is actually rather different in eukaryotic organisms, where the Kozak sequence as well as length of 5' UTR define the gene expression, and codon optimization is really organism specific depending on the abundance of tRNAs in the cells. In addition, eukaryotic gene expression regulation is much more complex because we have, beside the 3 universal box, we have also the transcription factors that can be multiplied and bound to the same gene, and then we also have a varying 3' UTRs with all concepts of regulation through 3' UTRs. Beyond that, we also have transcriptional enhancers as well as epigenetic factors, and processing of mRNA from the gene can be rather different. So splicing efficiency can be utilized for gene regulation, and at the end, as I said, for the post-translational gene regulation, the regulation of protein stability is rather different between the bacterial gene expression and eukaryotic gene expression. So we can say that classical scheme of gene regulation is really not uniform between all the organisms. So how we can overcome this problem? So what if we only focus on translation? So the translation is a process that basically translates the information that was given from transcribed from DNA to RNA into the protein, and it can be divided into three processes, which is initiation, elongation, and termination. When we focus on current research, we find that basically we heavily utilize various cis and trans regulatory elements that we are using for the control of gene expression. However, most of these cis and trans regulatory elements are rather, again, organism specific, and they really are focused on either on 5' untranslated region or 3' untranslated region of mRNAs. So we really wanted to focus on one step in translation that is really not that much studied in concept of gene regulation, which is translation elongation. And the reason for this is actually this step is the most conserved step between the different organisms. And if you can see here from the review by Marat Yusupov lab from a couple of years ago, the most conserved step in translation cycle between the bacteria and eukaryotes is elongation, depicted by this uniformed part in the middle, and as well as by similarity between the proteins involved in this cycle. So if we actually focus on translation elongation, we are wondering how we would actually be able to control this step in translation. Indeed, translation is a continuous cycle where basically ribosomes will bind to mRNA and then make from mRNA molecule some amount of proteins. If you think about elongation as a determining step in the gene expression, you could imagine something that would impede the speed of the ribosomes, so-called concept of ribosome stalling, and this would reduce the amount of the protein that is coming from the single mRNA sequence. This as a concept would actually be possible to utilize for the gene regulation. So our lab, including all the other labs in the world, show that ribosome stalling and pausing of the ribosome during translation cycle can really influence gene outputs. And shown in this schematic is how the encountering of stalling sequence will result in the production of the protein. So the ribosomes would start making the protein, and when they encounter the pausing sequence, they would stop or pause during this elongation cycle, and this would be a signal for the mRNA and protein associated factors and ribosome associated factors to start degrading the nascent polypeptide chain by ubiquitination, as well as cleave mRNA that is paused on the ribosome. This will result actually in the probability of ribosome passing through this stall or targeting of the message by these two mechanisms. And polypeptide chain that is being made up to that point would be further degraded by proteasome, while the mRNA that is sliced in the place of the pause would be degraded by further degraded by mRNA decay machinery. If we think about this in the concept of gene regulation, we would actually think that this way of gene regulation is perfect for the establishing gene dosage effects, because the amounts of RNA that would be transcribed from DNA would be really dependent on only on translation, how much RNA and how much protein would be in the system. And one should think about this as maybe non-efficient way of controlling gene expression, but it would be a great way of establishing gene dosage effects. Now, when we think about that, our lab and lab of Rachel Green over the last couple of years actually found one way to actually do this in really in the regulated fashion. So we actually found that the runs of polyA nucleotides in the coding sequences of the genes can actually regulate process of gene expression in a very linear way, as shown here either by the Western blot of cherry reporter with introduction of 9 to 13 A's in a row in the coding sequence of this gene. And as you can see from the two lower panels where we are showing the amounts of proteins or amount of mRNAs, introduction of coding sequences that have 9 to 13 A's in a row really reduce amount of the proteins as well as RNA stability in a very programmable fashion, going from 100% of the wild type in the wild type sequence to basically around 50% with introduction of 12 A's or 3 lysines that code for 3 lysines in this case. Now, over last 2 years, we actually showed that this is a concept that a lot of endogenous genes can are regulated with, as shown from the left panel, as well as we actually created a database of all sequences in the eukaryotic genomes that actually regulate endogenous genes in this way. We also showed what's the mechanism of regulation by this these, how we call them, polyA tracks, which is basically a concept of ribosome stalling and frame shifting on polyA track sequences. Now, when we actually found the mechanism and we found that this is the way how endogenous genes can be regulated, we actually wondered, can we actually use this method, system of gene regulation to actually create a tool that would allow us to regulate any gene in the cell. And we created this concept of polyA tracks for generation of hypomorphic mutants that is really dependent on the length of polyA track insertion in any given gene. Here shown by either reporter genes or in a concept of endogenous genes which has several exons. The one premise of this regulation was that increasing the length of polyA track, as I showed in 2 slides ago, is that we would be able to reduce the amount of the protein. We were also wondering whether this can be used in all the organisms, because again, as I pointed earlier, translation elongation cycle is the most conserved part of the translation cycle in the cells. So we introduced the polyA tracks in a reporter that we could express in E. coli cells, and as a control we used very similar reporter with lysine sequences that mimic basically polyA tracks, but they don't have a polyA tracks, but rather repetitions of AAG codons. And as you can see from the our Western blot or from the normalized fluorescence recordings, the polyA tracks were able to basically linearly, almost linearly, reduce the amount of mCherry reporter in E. coli cells, while the control reporters with AAG insertions were basically as a wild type level. So this already showed us that we could probably use polyA tracks for controlling gene expression in E. coli. So we went further from E. coli and we actually assayed the same kind of reporters in a tetrahymenate Thermophila, which is an AT-rich organism, and we were wondering whether we can use this method as well. As you can see from the panels in the lower part of this slide, we were able to actually again repeat what we did in E. coli, and introduction of 6, 9, or 12 repetitions of AA codons were able to reduce in a programmable amount of the reporter gene in tetrahymena. We also tried this concept in tissue cultures, in human tissue cultures as well as in the plant tissue cultures, and we found again the same method can be done in either plant, shown here in this slide, and this is tobacco-like plant, that basically served us to show that M-cherry reporters with introduction of AAA codons and introducing the polyA tracks of longer length would in programmable way reduce the protein amounts. These protein amounts are again very various hypomorphic basically alleles, create various hypomorphic alleles of the reporter sequence. So this was great, and we wanted to put additional step to this, which is basically, can we actually do this in a complete model organism? So we basically put our reporters in the whole fly larvae, and in this case we expressed it in the background of another reporter that is not controlled. So you can see on the left panel that we actually were assaying 3 different organs, but essentially the whole larvae was basically expression of the M-cherry reporter, and the whole larvae was controlled in this way. I'm showing you on the right panel currently expression of M-cherry reporter in salivary glands of this Drosophila larvae, but we also assayed it in the brain as well as in the proventriculus part. Again, showing that the concept of polyA track gene regulation in the model organism can be used on uniformly in all organs and all cells of this organism. This is actually an interesting concept because that would mean that polyA tracks are independent, regulating gene independently of transcription or amount of transcription, and that's what we wanted to test in a rather more controllable system, which is a human tissue culture system, and basically using a dose-dependent transcription using a doxycycline-induced CMV promoter. In this case, we also found that no matter how much we actually dose the transcription of our wild-type M-cherry reporter or M-cherry reporter with insertion of polyA track, in this case 36 A's or 12 AAA codons, we found that relative protein amounts between the wild-type and the polyA track gene were maintained no matter how much doxycycline we used to boost transcription of this gene, as well as RNA levels, which were basically staying to between 2 to 4% of the wild-type levels. At the end, we really wondered whether the insertion of 12 lysine residues would actually impede the function of the protein. So we turned again to the E. coli cells and inserted 10 lysine residues coded by AAG codon, or increasing amount of lysine codons coded by AAA codons, in this case increasing the length of polyA tracks into the chloramphenicol resistance genes. As you can see from this slide, where we actually assayed the amount of E. coli cells that can grow on the different amounts of chloramphenicol, the polylysine stretch of 10 AAG codons really didn't impede the function of the protein. However, in increasing length of polyA tracks that can be seen from AA3 to AA10, meaning from 9 to 30 A's in a row, had really like programmable reduction in the amount of chloramphenicol resistant gene that was actually seen in this figure as reduction in amount of the cells that can grow on increasing amount of chloramphenicol in the media. We also tested this in eukaryotic model, where we actually in the deletion strain of adenine biosynthesis pathway, shown as here's an empty vector that can basically not grow, the yeast that cannot grow on the minimal medium that is lacking adenine and can grow on the medium that is fully supplemented with adenine. We assayed how introduction of a single copy of either wild-type gene or gene that actually has engineered different lengths of polyA tracks would be actually seen as a functional readout. So as you can see, either here by generation of this red color that comes from the incomplete synthesis of adenine biosynthesis pathway, on the rich media we can see that introduction of longer polyA tracks is increasing the red color, which is again determinant of lacking of the AD1 gene in these yeast. And again, functionally, we could assay this by growing this yeast on the minimal media, where the missing gene or reduction in the gene into programmable ways was actually showing that the yeast that are lacking or producing small amounts of these genes cannot actually survive in the media without adenine. Now, this is an interesting concept that basically we would like to explore in other systems, and currently we are trying to move this into the more problematic systems. But I want just to summarize what I told you up to now and basically show the difference between the polyA tracks and classical gene knockout techniques. So the classical gene knockout techniques would basically reduce the amount of genes by targeting either one or both alleles of genes in diploid organism, which would actually be a reduction of 100% in the wild type to 50% in one allele loss, which again, in the problem in haploinsufficient genes, these animals or these genes would basically influence survivability of the organisms even at the loss of 50%. Again, in most of the study, complete loss of the genes, meaning missing both alleles, would usually lead to death if the gene is essential. With the polyA track method, we can actually now tune in basically any amount between 100% to basically 1% or 0% of wild type, and we can actually study the effects of haploinsufficient genes basically by creating 85%, 75%, or any amount above the 50%, which would correspond to a one allele loss. And then again, we can actually study effects of all the reductions in the gene amounts from basically 50% to basically 1%, again by using a different polyA track that would reduce the genes levels from basically 50% to 1% of the wild type. With this, I would like to basically finish my webinar, and I would just like to acknowledge people that contributed this work, mainly PhD student Laura Arthur and the research faculty in the lab Slavica Plavlovic-Jaranovic, as well as our collaborators from Poland, Pavel Szczesny, Johns Hopkins, Rachel Green, Washington University here in St. Louis, Heather True Krob, Douglas Joker, Jim Skeet, as well as our collaborators from Canada and University of Western Ontario, Vava Gurbic and Rima Menez. And I would like to thank you for your time, and I would like to take any questions about our study. Thank you.
Thank you, Sergey, for such an interesting webinar. It was very educational, and I'm sure listeners enjoyed the presentation as well. We had some questions come in throughout the webinar, and we will try to address as many as we can. So just to quickly get started with the questions, the first question, which seems to be a pretty broad question, but I think it is an interesting one as well. What kind of research areas could most benefit from such a sensitive gene expression tool?
Hi, Sumit. As we wrote in our article, I think that basically gene expression tool, as we actually envisioned it, can help in several research areas, but we actually went with synthetic biology as one of the most, I would say, applicable research areas where we could apply our gene expression tool. However, it's not only synthetic biology that is important for where the very precise control of gene expression is important. We envision that this will be used in any industrial area where you would like to optimize production of certain secondary metabolites or antibiotics, even in the recombinant antibodies with now immune therapy. You need to basically optimize heavy and light chain production, and any basically area where you want to this pathways where you have several genes, you could basically tune in expression levels of any gene by using this tool. Obviously, big area where we would like to actually apply this is diagnostics, where we want to basically, as I talked in my presentation, where we want to basically assay now how certain genes like tumor suppressor genes and oncogenes react, or how the cells react when they are attenuated to a certain level. So we envision it in basically any industrial areas, but also as a part of the research tools for anything that is connected to diseases that are dependent on the certain changes in gene expression levels.
Okay, great. Thank you very much for that. And the second question, and again, this is a pretty broad question. So what are the next steps towards a therapeutic solution for diseases related to partial gene loss function?
So as I said in my presentation, and as I just mentioned in answering your first question, what we think basically how to use this tool basically as a research tool is basically now we can actually engineer any of the gene that we are basically thinking is involved in diseases, and basically see how the reduction or partial reduction of expression in that gene is actually influencing the cell homeostasis. So what was missing before and what was mentioned in several articles explaining our work was basically this tunable basically reduction in the gene expression, because genetic tools that were used before were either using complete knockouts or single allele knockouts. Using siRNAs, we could basically tune in things, but not all the organisms and not all cells will react well on transfection of siRNAs. And now we can basically do this for any organism, basically, and we could do it basically in any cell, and we can basically tune to any certain value that is reduction from basically, as I said, from 1-2% to basically 99% of wild type expression levels.
Yeah, that's pretty impressive. And just for the interest of time, we will take one last question. And this question is more about, during your presentation, you showed that adding polyAs effectively reduces gene expression. By the same token, does removing polyAs increase gene production?
Yeah, so we explained that in a previous publication that basically there are genes already in human cells that have polyA tracks. There is around 450 genes, approximately 2% actually of any organisms will have polyA track genes. So 2% of the genes are with endogenous polyA tracks. In these genes, actually, synonymous mutations, so actually, as you are implying, removing polyAs are actually shortening them or basically changing that lysine codon from AAA to AAG synonymously, meaning not changing the protein, would actually have effects on the expression of the genes. And as you envision, actually adding Gs in the polyA tracks without even changing the length of polylysine stretch that is coded by that will increase actually expression of that gene. And vice versa, if you have a G already, because we made a polyA track as something that has 12 A's interrupted by one nucleotide at any position, removing one of that nucleotide or basically changing it to an A will actually reduce expression of that gene in endogenous context. So you have a possibility for endogenous genes to go even to a higher expression or a lower expression.
Wow, that's very interesting, and I think a lot of people can actually probably use this approach for a better protein expression as well at that point. So
yes,
So this is all the time we have today. I would definitely like to thank Sergey for presenting as part of our GeneWiz Week webinar series. Along with our webinar series, we have other ongoing activities such as Lab Startup Program, GeneWiz Credit Awards, and exclusive promotions. Make sure you check out genewizweek.com. Our next webinar will be held today at 5 p.m. Eastern Standard Time, featuring our own Edward Messick from GeneWiz. He will be presenting on metagenomics for biomarker discovery, applications in precision medicine. All of our webinars will be recorded, so even if you can't make it, please register and we will send you a link. Thank you very much for attending this webinar, and you have a great day. And thank you, Sergey, once again.
Thank you, Sumit.
Bye.